And the beauty of the light therapy, the infrared light therapy that we developed, or we developed the idea of using, and it's a specific wavelength. That's the first thing to bear in mind. The wavelength we use is around the 1070 nanometers, which actually 20 years ago was thought to do nothing biologically. And therefore, we actually had to convince a company to make the LEDs to use it. And when the reason we chose 1070 is because of that range of wavelengths, was that it penetrates the brain. The first thing is you need to understand when you're a drug developer or a therapeutic developer, your therapy has to reach the targets where the problem is in the brain. And the brain is quite obviously, it's a structure underneath a skull, and it's quite large, and you have to penetrate. And light, again, people think how can light possibly penetrate tissue, but actually the infrared ranges can penetrate quite significantly, and the wavelength we've adopted and used and studied for many years is the most penetrating. It turns out to be the most penetrating wavelength in the infrared range, and that's a good thing. So obviously that suggests that it will get to your target where you want to treat a disease. And the other important thing of this wavelength is the peak wavelength for transmission through water. And water makes up 70% of your brain. So once it's in, it actually travels further into the brain because of that fact. And that's the reason we chose that wavelength initially. But as it turns out, okay, it gets to target, but also it does all the things we wanted a therapy to do in the brain. It does pretty much everything we've just described for the disease, like Alzheimer's. It's an antioxidant, anti-inflammatory. It actually targets this system that clears unwanted proteins. So one of its targets is a channel that controls that process in the lymphatic system. So that's one of the advantages of this wavelength is actually it targets that as well as the other major target, which is the mitochondria. So what it does, it boosts mitochondrial function, which is well known for many of the wavelengths of infrared light. It boosts ATP production. It boosts a very important thing. It actually increases a little bit of nitric oxide, and this is a gas. You might think it's a bit strange, a gas, nitric oxide, but actually nitric oxide is an incredibly important molecule in your brain and in your body generally. It actually controls whether your blood vessels dilate or not. So what it does, it relaxes a blood vessel and increases your blood flow in the body. And that's what our light, the light therapy does, the infrared light therapy. Many of the wavelengths do this, actually increases a bit of nitric oxide. So locally, your blood supply is much stronger. So you get more blood flow, more oxygen to the brain, which is very important in aging. It's very important in disease, brain disease. Because again, in aging, your blood supply, starts to wane a bit. So you don't get so much oxygen into your brain, which is not a good thing because the brain neurons need oxygen. In fact, they use about 35–40% of the total oxygen in your body because they are very what we call excitable cells. They need to function and be excitable, and you need energy to do that. So ATP and energy production is incredibly important. And fortunately, like the infrared light therapy does give you that extra blood supply. It actually increases oxygenated hemoglobin, which is the very important carrier of oxygen to your brain. And again, 1070 seems to be the optimum wavelength for doing that. It gives you a longer extent of oxygenated blood to your brain, which is all good stuff all good news for aging. It's all good news for age-related diseases where it starts to deteriorate. So yes, so as well as that, one interesting thing also is it also in the mitochondria, what it does, it produces a little bit of what we call reactive oxygen species, which most people will say, oh, that's not a good thing. This is an oxidative stress molecule. But actually a little bit of oxidative stress is actually a good thing. What it does, it triggers your body's system to protect your body. So it's what we call preconditioning. And the light appears to do this. Is, I call it sort of photo preconditioning. So it does produce a little bit of reactive oxygen species, which then causes transcription of key protective proteins, and these are called chaperone proteins. So we've shown with our wavelength, again, in an Alzheimer mouse model, that you can actually produce, you increase these so-called chaperone proteins. And these chaperone proteins are really important for protecting your cells from this stress that I've talked about all the way through, all these stressors. So it protects against oxidative stress. It helps with, again, with the protein stress, the misfolded protein stress. And also, it processes those misfolded proteins. And but the biggest result we found a number of years ago, what I call a fall off the seat moment, was actually treating the cells of our light, increased the chaperone proteins, but at the same time, what it did, it reduced amyloid, one of the key proteins in Alzheimer's disease, down by 80-odd%. It was a massive reduction in amyloid. And it wasn't just the amyloid that you find on the outside of a cell. It's actually the key amyloid you need to target inside a cell that's killing the cell ultimately, was reduced by this therapy. And also we found the other key pathological protein, phospho-tau, was again reduced by this treatment. And we think it's because of this preconditioning process that the chaperone proteins are reducing these unwanted proteins that are in the cell. And that was obviously a really exciting observation in our preclinical studies, that that you could have a treatment that would actually reduce the key pathological proteins within Alzheimer's. And that led to the first trial, actually in humans, the idea that we could actually potentially use it in humans for treating Alzheimer's disease. Okay, so it's a combination of things. It's the all the stresses we talked about, it combats, and it also importantly reduces some of the pathological proteins as well, and no side effects. That's, again, I have to emphasize.
Because I did actually try to find any devices in the 1070 range, and I think I found like one company, but the vast majority of devices that are available commercially are like 810, 850.
Yeah.
What difference do you think? Can you comment on whether there would be any what differences you would expect?
Yeah. So just the first point, so I mean, there are two companies basically that you can get the wavelength, including my original clinical colleague that knocked on my door 20 years ago. That was used for the trials I've been talking about, actually. Anyway, but actually some companies are starting to look at 1070. But going back to your original question, the wavelength, the two main wavelengths that I think are most useful are ours, obviously, but also around the 810 nanometers. And the main one of the reasons is, first of all, it penetrates, similar to the 1070 into the brain, so it gets the target pretty much equally to the 1070 wavelength. So that's the first point. And also what it does, that particular wavelength, yes, I would say is potentially it is very useful. There is clinical evidence for its use, and it does similar things to our wavelength. So around about the 810, I would say is a very useful wavelength. In fact, we're very keen to compare it. We're currently working on comparing the two wavelengths, and we are working with a company that has worked with the wavelength a lot. And in fact, that company also is looking at exploring combining 1070 with 810 in a dual system, and it's doing it actually quite cleverly, because it may be that you need to target one of the wavelengths to a certain part of the brain, whereas the other one potentially is more useful subcortically versus cortically. So that's been looked at, and what's this space? Basically that will be available, and there's some papers coming out on that idea of doing a combination of the two wavelengths in two different parts of the brain. Actually, the other thing I haven't mentioned actually is the physiological effects of this treatment. A lot of EEG has been done in the brains of people that have been treated. Again, you see modifications and plasticity of the brain. The physiology of the brain changes positively with the treatments, and both 810 and 1070 do similar things. So I would say 810, all in all, from the evidence, is as useful as ours. The only thing is what we don't know, because the research hasn't been done, is whether the 810 also targets this lymphatic target I mentioned. So it certainly modifies the mitochondria, boosts ATP, oxygenated blood, all the things we've been talking about. Nobody studied the chaperone effects, which we observed. That's another thing we don't know that the 810 does. My guess is it will do similar things. But also, as I said, we don't know if the 810 actually modifies this lymphatic effect, which I think is incredibly important. And it's a sort of benefit of the wavelength we study. It targets the mitochondria, and it does this lymphatic process. And you need both really to give you maximum benefit in a disease like Alzheimer's. So yes, in answer to your question, 810 is a good penetrating wavelength. It does many of the things that the 1070 does. There's still some research that needs to be done that it does the full complement, if you like, that we've seen with our wavelength. So there are some successful clinical trials. There's small clinical trials I've seen with 810. Mostly, a lot of them are case studies, single studies rather than full studies. Like we've done a couple of full studies. So yes, 810 is fine. There are other wavelengths, and there are other companies that are selling devices that give you other wavelengths, shorter wavelengths. Again, those wavelengths have some logic, in that they again will do a lot of what we talked about in terms of mechanisms, positive mechanisms, particularly on the mitochondria effect. The only problem for me is penetration. The shorter wavelengths really don't penetrate anywhere near into the brain. So they're useful for surface effects, maybe effects on the blood supply locally, and there is again, your viewers may not be aware, but actually it's recently found that you can find mitochondria, those batteries I talked about in cells, you can find them in the blood supply. So there's mobile mitochondria that basically travel around your body. So there is a possibility you can treat with these shorter wavelengths that can modify the activity of these mitochondria that then can get to places in the brain. That's a possibility. As I said, I'm confident with the 810 region. Again, there's a lot of they use 850. Again, don't get hung up on wavelengths. This when people say 810 or 808 or 820, and we talk about 1070, 1072, 1064, they're all the same basically. The thing is, people have just to give you some insight, there's an issue about patenting these devices and wavelengths. There's a lot of, they manipulate the number. People call it 808, 810 to avoid being issues of patent, issues of IP and things like that. So we've had to do similar things. Our original wavelength was 1072, and then we shifted it to 1070 and then 1068 just to avoid issues, problems. I mean, it's very strange, and people,
Yeah.
the general public, don't realize it's basically the same wavelength. Don't get hung up on, oh, 808 is different to 810. It's a number. It's plus and minus 10 nanometers, 10, 15 nanometers is basically the same wavelength doing the same thing mechanistically. And there's a common misconception. I get asked a lot about that. And also there's things about, again, some devices give you a constant light, whereas we think pulsing, it might be a question you might be asking me next, pulsing is important. So pulsed light and also the power of the light for penetration is important. So again, you get variability within companies and devices in terms of that as well. And again, using LED or lasers, again, we favor LEDs more on a safety side of things. But saying that, people had sold devices with lasers, and they have published reasonable evidence for use. So there's things like that. So pulsing is important, and the power. So you need a certain degree of power as well as for penetration. And if you haven't got enough power, really, again, your penetration is limited. So and pulsing we think is important and not repetitive. Right from the beginning, when we did our preclinical studies, repetitive treatments is important, not just a single treatment. All our preclinical was a series of short but repetitive treatments seemed to be beneficial to the process, to give you sort of the best results, if you like. And we do similar things, obviously when we're treating humans, it's twice a day, 6 minutes, short exposure, twice a day, 6 to 10 minutes, every day. Again, it's repetitive treatments seems to give, well, it gave us the results we got. I wouldn't say, I can't claim that that's optimal by any means. We haven't had the finances to basically to do all the experiments, basically to demonstrate that we have reached optimal levels of treatment.
Dr. Shazo provided a few details for the light used in the studies that he confirmed after the call. In the interview, he mentioned a wavelength of 1070 nanometers. To confirm, the wavelengths used were from 1060 to 1080, and as he refers to in the interview, the exact wavelength may not be that important and is mostly driven by IP issues. The overall power of the device is 15,000 milliwatts, which is 15 watts. This gives a power density on the skin of 23.1 milliwatts per centimeter squared, across an area of 650 centimeters squared. The device does sit on top of the head, so is very close to the surface. Something I've been really digging into recently is how we can protect our DNA as we get older. And there has been some fascinating research coming out of Australia about magnesium. They found that when we're low on magnesium, it leaves our DNA more vulnerable to damage. Meanwhile, up to 80% of us are walking around with low magnesium levels. To maintain optimal levels, I supplement with magnesium, and I specifically choose Magnesium Breakthrough by Bioptimizers. What sets it apart is its comprehensive formula, containing all 7 highly bioavailable forms. And while the magnesium is protecting my DNA, I've also noticed I'm sleeping better, feeling less stressed, getting better recovery after workouts, and have more energy. If you want to try out Magnesium Breakthrough, they have a 365-day money-back guarantee. And by visiting bioptimizers.com/modern and using the code MODERN10, you will get a 10% discount. If you subscribe, not only will you get amazing discounts and free gifts, you will make sure your monthly supply is guaranteed.
So when we look at, of course, mitochondrial activity, we're specifically looking at what is actually the acceptor of near-infrared light. In this case, we use 1070 nanometers. So I know there's other types of, within the photobiomodulation industry, we've got 810 nanometers and red light therapy of 600 nanometers. but they all, in relative perspectives, do similar things. They all activate a protein structure called the cytochrome C oxidase. And what we're really trying to do there is support mitochondrial activity, or support the rate of electron transfer of the cytochrome C oxidase. If we increase or support the rate of cytochrome C oxidase, we can improve and support at least the rate of mitochondrial activity.
Okay, so I do want to talk about the the wavelengths in a second, but it also increases the blood flow, and I haven't heard that before. Do we know why it increases the blood flow? Is it just because the mitochondria need more, I guess, food to generate this energy, or is there some other process?
So within the cytochrome C oxidase, you also have nitric oxide, and the light can displace nitric oxide within cytochrome C oxidase. And when it's displaced, it can flow through into your vessels, and of course vasodilation can occur. And with this, and with the increased rate of ATP reduction, you can, in this sense, increase the rate of cerebral blood flow. And if we're talking about transcranial fault biomodulation, for instance.
Okay, yeah, that makes sense. So it's releasing the nitric oxide, which is causing the vasodilation. Okay, excellent. So as you mentioned, many other companies or many other systems use different wavelengths, but you have chosen 1070, which is actually quite a rare a rare wavelength. Why 1070? What are the benefits of 1070 as compared to the other wavelengths?
Yeah, so we're talking about penetration depth, certainly. So the longer the wavelength, technically, the longer or further light can penetrate up into a degree. Then you've got the absorption of light through water molecules, and then after about 1080 nanometers, it just cuts off, and light can't actually penetrate deeply. But there's that sweet spot almost with 1064 nanometers, 1070 nanometers, where you can actually get good penetration depth because the light scattering coefficient is actually reduced compared to higher energy or shorter wavelengths, such as 810 nanometers and 660 nanometers. And because of this penetration, you can increase or support a greater volume of cells or mitochondria.
Yeah, I think another one that was mentioned was there was a study with Hanley Liu and Tyrrell Pruitt, and in that study they showed the use of 810, 850, and 1064 nanometer lasers, and they used lasers primarily, and then they checked alongside with LEDs. And in this study, they actually showed that the 1064 had longer penetration, but actually longer sustained effect as well. And so that's why we really chose this wavelength.
Right. It's interesting about the light absorption, because we did talk to someone who said anything over 1,000 gets, by water, that it gets absorbed by water. But if you're just below this 1080, then you're okay. Basically, it doesn't get absorbed.
And I think actually there was an interesting presentation done by Dr. Paul Chazot in Durham University, and he wasn't saying anything about actually the water stopping the light getting through, but actually as a magnifying glass, similar to how like a magnifying glass would heat up and concentrate the sun's light on things. He was saying the same thing would happen actually with the 1064. Yeah, and the interesting thing about light therapy is the biphasic dose response. And this was something that came up at the photobiomodulation conference in London last year, and it was reported on. But really, there is like the Goldilocks principle where you can have too much or too little, and it's different for everyone. Some people are very sensitive to external stimuli. Some people will get dehydrated in the sun a lot quicker than others, and so we have to work sometimes with people to figure out what that sweet spot is for them. And that's why sometimes a consultation is very helpful.
So what kind of symptoms would you get if you were overdoing it?
You might feel like, similar to been on the beach for too long, a little dehydrated, where you might seem a bit like drowsy or fatigue. Typically this doesn't happen with sessions when you're using it, any of the preset protocols or the custom ones that we develop. But as Will said, if you're starting to kind of do things over 30 minutes and kind of take matters into your own hand, then you might just feel like, tiredness after using it, that your brain is just being overused because it's firing too much. So that kind of a way.
So I've got light coming in and it's changing the protein in the mitochondria. Does it have to be at like a specific wavelength to be able to like excite electrons or whatever to make that change? Or will anything, or how wide can the wavelength range be?
I think we're pinning that one down quite well. It's the longer wavelengths of light. So we'll say red into infrared. In terms of numbers, it's about 660 nanometers, which you would see as quite deep red, through to 900 nanometers, which you can't see. But once you've gone beyond about 750, 720 nanometers, you can't see the light anymore. If you turn the energy up a lot, you can just see a dim red light. But those are the wavelengths that are important. I think there are a few hotspots in those wavelengths. However, anything in those wavelengths will have impact. Our solar environment, solar light, is particularly rich in those wavelengths, but we ignore them because we can't see them. Our visual system can't see them. And that you tend to ignore what you can't see. And that has been a bit of an issue. So yes, it's sitting under, I don't know, a fluorescent tube is going to do nothing for you. Going outside, you're going to get a lot of infrared light. And if you go back to our old incandescent light bulbs again, you'll get lots of infrared light.
Right. Okay. And kind of anything in that area can have this positive effect.
As far as we know, yes, it can. As I say, I think there are a few hotspots, but the trouble is you could waste half your life going nanometer by nanometer by nanometer trying to find out exactly what is the hottest spot there. I think if you think about evolution, we've evolved under solar light, which has got this very, very broad spectrum. The infrared in solar light goes up to about 2,500 nanometers. That's probably not much good to us, because when you go over 900 nanometers, most of that light will be absorbed by water, not by the biology in your body. So what we're looking at is really 650, 660 through to 900 max. In the past, everyone has used 670 nanometer light. Now, there is no reason for that. The only reason is the first people that did the experiments, or started on these experiments, which were really a great 2 or 3 groups in Australia, happened to have a device which produced 670 nanometers. So that became a benchmark. So we know a lot about what 670 nanometers does, but you can do almost exactly the same with 850 nanometers, which you can't see. So I'm not going to pull that spectral range apart. It's too much work for very, very little reward. Let's just make sure we get a broad spectrum of infrared light in our lives.
How deep does 670 go? Yeah, I mean, can you get to your liver?
Oh yeah, it certainly can get to your liver. It won't come out the other side. It's not quite long enough to do that. But 670 will certainly go through your skull. So for instance, in looking at aging, we have used 670 on the eye to improve vision in people that are older people, very successfully. And it transpires that when we do that, we're also illuminating the inner ear. So, it is going through, longer wavelengths go through bone. That you automatically think it's bone, that's going to stop it. No, it doesn't. Longer wavelengths will go through bone, and you can barely see the bone as a very, very distant shadow. But the 670 will not go all the way through the body. Right. But it will certainly go pretty deep. I mean, I strongly suspect in fact, we've argued this before, that because your chest is vacuous, it's largely an empty space, we can use 670 to get to the heart. So one of the arguments that I think has a lot of traction is in terms of things like if you have a heart attack and your cardiac tissue becomes ischemic, right, doesn't have enough oxygen, we know that 670 nanometer light improves the ischemic situation. So I would love to see a 670 light actually in an A&E unit. And when people come in with an ischemic crisis, I mean, we know from all the science that the red light and 670 in particular can help ischemia. You know, it costs nothing. It's going to do you absolutely no harm. And if it saves 1 in 1,000 people, that's a winner, as far as I'm concerned.
Yes.
So, yeah, different wavelengths. Longer the wavelength, the greater the penetration.
Okay, if we think about dosing, so it's like power and time, or are there other parameters? How do you, when you're doing an experiment, how do you I guess calculate what dose you will use?
That's a terribly good question, and it's also an extremely demanding question, because with the red light research, we have too many variables, so we can't isolate one variable very easily on its own. You have wavelength, you have energy, you have time, and you have time of day, because your response to red light changes at different times of the day. So how do we manage that? Well, I don't have access to enough subjects to pull that one apart. We've done a lot of experiments on animals, and what you find in this is highly conserved in evolution. What you find in a fly, a bumblebee, or a human is exactly the same in terms of red light metrics. So we know that if you go below around 1 minute, 1 minute 15 seconds is the break point. If you give red light at any energy below that time period, nothing will happen. And it's not like aspirin. Take 5 aspirin and your headache will go away quicker than if you take 1 aspirin. It is something that we now know is a switch. When you do something, you turn the switch that improves mitochondrial function, and that switch, once you've turned it, will last 5 days. Again, doesn't matter if you're a fly, a human, whatever. Now, physiologists find this very difficult because in physiology, there aren't a lot of switches. There are a lot of dose relationships. So when we've done work on aging people and aging eyes, and we've managed to improve vision, we say to people 3 minutes in the morning. We know 3 minutes is an overkill completely. But then if you've got someone that tends to chisel things away, so you say to them, oh, it's 1 minute and 15 seconds, someone's going to do a minute. Someone's going to chisel it away. And so for that reason, we've gone over the top and we say, do it for 3 minutes. Now, when this scenario came out, people had a fixed energy for using this, and it was 40 milliwatts per centimeter squared. Bit bright. So what we did do is we started reducing that and still looking for an effect. And so we had a project in Moorfields Eye Hospital where we took some people and we just reduced the energy. And so we can now deliver an effective dose. Again, remember, it's a switch at a much lower energy and get as good a physiological effect as if we give high energy. So we can now deliver something that is very comfortable. And no one's going to, we've got clinical trials with young children. We've got trials with older people, and they tolerate it very, very easily. We're down to about 5 milliwatts per centimeter squared, which is, if you don't know what that looks like, it's a relatively dim bicycle light.